Hey, Mr. P here. This is lecture 5 of chapter 8. This will be the final chapter 8 video. And in this video, we're going to talk about the Calvin cycle, which is also called the light-independent reactions or the dark reactions. And I don't particularly like to call it the dark reactions, only because dark reactions kind of implies that it requires the dark in order to work. And it doesn't require dark. It definitely can happen in the dark and take place in the dark, but it equally can happen in the light. It's just called the light-independent reactions because they are completely independent of light, meaning you do not need or require light in order for the Calvin cycle to take place. This is a good kind of overview of the light-dependent and light-independent reactions. It's the good overview of photosynthesis. Notice that the light reactions, which are basically this half, okay, light-dependent reactions, are the first half of photosynthesis, and the second half of photosynthesis would be the light-independent reactions. Okay, we've got the light-dependent reactions and the light-independent reactions. In the light-dependent reactions, remember we're going to use H2O. Specifically, we're going to break down water. That is a specific oxidation reaction. Remember, oxidation is losing electrons, and water will be split up into three things: H plus ions, electrons, and O, or half of an O2 molecule. Those three things will be put into the light reactions, which is that photosystem 2, photosystem 1, electron transport chain, chemiosmosis portion of photosynthesis. Oxygen will be our byproduct. Oxygen is going to be released to the atmosphere. And this process requires an input of light. Remember, photosystem 2 and photosystem 1 are going to include chlorophylls, and those chlorophylls are going to take the photons and will eventually energize electrons at those photosystems, specifically P680 in photosystem 2 and P700 in photosystem 1. But that light energy, which is again why this is called the light-dependent reactions, is going to split water and energize electrons. Now, those energized electrons are going to do work to maintain that H plus ion concentration gradient, but they will eventually be accepted by our final electron acceptor, which is NADP+. NADP+ will accept the electrons and a hydrogen ion, be converted to NADPH, and NADPH is our final electron acceptor, full taxi, which is going to obviously drop 2 electrons and 2 hydrogen ions at the Calvin cycle. Okay, so NADPH is going to take hydrogen ions and electrons from the light-dependent reactions into the Calvin cycle. ATP is another thing that's produced by the light reactions. ATP is produced by that chemiosmosis portion. ATP is obviously going to be the energy source. I'll just put an E. That is the energy source for the Calvin cycle. NADP+ will be produced from the Calvin cycle, because when NADPH drops off those electrons and hydrogen ions, it eventually is reduced or oxidized back to NADP+, and so it's just like a taxi taking passengers from the light reactions. It'll drop off those passengers and return back to the light reactions as NADP+. ATP will drop off the energy and will be converted back to ADP plus inorganic phosphate. So notice that these two things are essentially the same two things here. The only difference is ATP, the energy that ATP is holding is going to be used, converting it back to ADP. NADPH will drop off the hydrogen ions and electrons and be converted back to NADP+, which will eventually return back to the light reactions. The whole purpose of the Calvin cycle is to fix carbon dioxide, which means basically remove the carbon from atmospheric carbon to an organic molecule carbon in sugar, and that sugar is glucose. Okay, glucose. We know glucose as C6H12O6, and so this glucose will be made from the carbon that is locked in the atmosphere, and essentially that's the Calvin cycle's job. Okay, the plant needs glucose, and so why does the plant take in carbon dioxide? Well, it's basically to fix carbon into an organic molecule form, And we know from our organic molecule unit that glucose, or any carbohydrate, is designed to store energy short term for the plant. So the plant will store glucose or store energy in glucose until it's ready to break that glucose down into ATP molecules. Okay, so let's talk a little bit about what the Calvin cycle is. And so here is our absolute, and maybe showing my age a little bit, but we have the dark side and we have the light side. And I like to think of this analogy in terms of, or when we think about photosynthesis, because you have kind of, in this case, in Star Wars, you have a division between the dark and light side, just like you have a division between the light reactions and dark reactions. The light side is obviously going to produce your NADP plus and your ATP, which are good molecules, and the dark side are going to take those good molecules and they're going to convert it into glucose with the inflow of carbon dioxide. And so you have little Yoda down here, which was always my favorite. And you know, he would say things like, "Oh, NADP plus and ATP. We like ATP." And then you have Darth Vader. "I want your energy." And so they would send their ATP and their NADP plus, and the dark side would eventually make glucose out of it. Anywho, Calvin cycle. Oh yeah. Get ready. Here it comes. Okay, so the dark reactions. We are going to start with step 1, and there are eventually, or essentially, 4 steps that you need to know. The first step is carbon fixation. And carbon fixation, just like it sounds, is we are fixing carbon. And so carbon is in the atmospheric version, which is CO2. Okay, carbon dioxide is a gas in our atmosphere. It's one of the greenhouse gases. It is one of the reasons why our planet is warming. But carbon dioxide is kind of a carbon sink that's in the atmosphere. Okay, there's a lot of atmospheric carbon out there, and plants' job is to fix that carbon into a version that the plant can use to store energy. The plant can't use CO2 to store energy, but it can convert or fix this version of carbon dioxide or of carbon into a version that the plant can use, which is glucose. Okay, so we're going to start. This is our starting molecule. We're going to start here, and it's a cycle, so we're going to start and end at the same place. So this is where we're starting. We're going to start with 3 molecules of RUBP. That's ribulose 1,5-bisphosphate, and you don't need to know the long version, the long name. You can call it Rubisco, and you can see that right here. It's called Rubisco, or you can just call it RUBP. Ribulose 1,5-bisphosphate is basically a ribulose with a phosphate attached to the first carbon and the 5th carbon. So that's ribulose 1,5-bi or bisphosphate. Okay, 2 phosphates. This RUBP is a molecule that we're going to start with, and we're going to add in 3 carbon dioxide molecules. Now, the reason we add 3 CO2 molecules is because we want a 6th carbon for this RUBP molecule. We have 5. 1, 2, 3, 4, 5. We have 5 carbons. We want to add a 6th carbon to our 5 carbon molecule, which is called RUBP. So we're going to add 3 molecules to our 3 5 carbon molecules, and you will get 3 6 carbon molecules. So 1, 2, 3, 4, 5, 6. On these, which are all bonded together, will be the phosphate on this side and the phosphate on this side. Okay, so we have now 3 6 carbon molecules with a phosphate attached to each side, which is basically this exact molecule, but we added a carbon to each one of these 3 5 carbon molecules. So as soon as you get 3 6 carbon molecules, you will split it in half, because the bonds that made these ribulose 1,5-bisphosphate or RUBP molecules aren't strong enough to support the weight that all of these things are causing on this middle bond, and so it eventually, or kind of instantly, breaks in half. As soon as you add this 6th carbon to this 5 carbon RUBP molecule, it splits it in half. And so now we have 6 molecules that each have 3 carbons and a phosphate. So that makes sense, right? We have 6 3 carbon molecules because we started with 3 6 carbon molecules. Once you split these 6 carbon molecules in half, we get 6 3 carbon molecules, and the name of these are 3-PGA. Okay, you have to absolutely know 3-PGA. The name of that is 3-phosphoglyceraldehyde or phosphoglycerate. These 3-phosphoglycerate molecules are going to be 3 carbon, 1, 2, 3 carbon with a phosphate attached. Okay, so that's the first step, carbon fixation. We take a Rubisco molecule, we add a carbon to it, and now we have twice as many 3 carbon molecules. That's phosphoglycerate. Second step is the energy consumption and the redox step. We know what reduction and oxidation reactions are. Redox reactions are when you have a reduction and an oxidation. Reduction, remember, is the gaining of electrons, and oxidation is the losing of electrons. We remember that by using the acronym oil rig. But what happens is once we have these 6 3 carbon molecules, which are called 3-phosphoglycerate, we are going to add 6 ATP molecules. Why 6? Because we need 1 ATP molecule per 3-PGA molecule. So if we have 6 of these, we need 6 ATPs. That means we're going to add energy to each one of the 3-PGA molecules. Okay? Where did this ATP come from? It came in from the light-dependent reaction. We just made that. That was chemiosmosis that made that. So it comes in, it drops off the energy, and it's going to return as ADP and phosphate. Okay, what did it drop off? It dropped off energy. Now we're going to bring in 6 NADPH molecules. And what are the NADPH molecules coming from? They're coming from the light-dependent reaction 2. That was the final electron acceptor. Remember, NADPH is a full taxi. It's going to drop off the 2 electrons and hydrogen ions. And so when the electrons and hydrogen ions, together with the energy that is dropped off by the ATP, join these PGA molecules, it's going to cause the 3-PGA molecules to turn into G3P. All we do is we add a hydrogen and electrons and a little bit energy to these 6 molecules to convert it into something else. Okay, and G3P stands for glyceraldehyde 3-phosphate. So it changes from 3-phosphoglycerate to basically 3 molecules of glyceraldehyde 3-phosphate. And all we're essentially doing is we're adding the energy and electrons and hydrogens to change where this phosphate actually bonds to. And if you want to be the most specific, you would say that this phosphate, instead of being bonded to the end, actually changes and bonds to the middle. But you don't necessarily need to go into that much depth. Once the molecules are changed into glyceraldehyde 3-phosphate, or G3P, we're going to go into the third step. And the third step is basically removal of a G3P. Okay, so we go from 6 to 5. And so how do you go from 6 of anything to 5 of anything? You have to take one out. And so step 3 is essentially the easiest step, I think. Step 1, carbon fixation. Step 2, redox. We are basically reducing these molecules by adding electrons and hydrogen ions to them. That means we're oxidizing this molecule. And then in step 3, we're just removing one of the G3Ps. We're going to hold on to that because 2 G3Ps makes a glucose. Okay, so if we pull one out, we're left with 5. And then step 4 is going to be basically regeneration of RUBP. And so how do we regenerate RUBP? We are going to add some energy. We're going to bring in 3 more ATPs. Why 3? Because we need to regenerate 3 RUBPs. And so do the math. If we start with 5 3-carbon molecules, we have 15 total carbons. We're going to add this energy to kind of reschedule, or not reschedule, but reorganize these 15 carbons that are now in 5 triplets into 3 5-carbon molecules, and the only way you do that is by adding energy, and so it's a simple step as well, right? We take 5 3-carbon molecules, we add some energy to shuffle the carbons around, and we come up with 3 5-carbon molecules, which is totally possible because there's 15 total carbons here, 15 total carbons here. It's like saying we have 15 total Legos. Instead of having 3 Legos bonded together 5 times, now we have 5 Legos bonded together 3 times. Okay, and that is the Calvin cycle. Okay. Notice that the ATP is used to rearrange Rubisco for each molecule made. Again, we need 3 ATP molecules because we are regenerating 3 RUBP molecules. All right. So I know these slides were kind of messy, and so I'm going to kind of simplify it, and we're going to look at this. It's the exact same thing. This one kind of breaks it down into 3 major steps. Notice that there was a 3rd step here, which we'll talk about. And so we being Mr. Hamilton, Mrs. Wickman, and I, are all 3 going to require or expect you to be able to draw this. And so I'm going through it one more time. Make sure you can go through it with me. First step is right here. First step is carbon fixation. And we are going to start with our 3 5-carbon molecules called Rubisco, or ribulose 1,5-bisphosphate, or shortened to 3 RUBPs. RUBP is going to be basically reduced by gaining electrons by this carbon fixation step. And so we bring in 3 CO2s. All we need is carbon dioxide because all we need from it is the carbon. And so we're going to take 3 carbons. We're going to add 1 carbon to each of the 3 RUBPs. And if you add 1 carbon to each of the 3 5-carbon molecules, you end with 3 6-carbon molecules. But what's the problem with the 6-carbon molecules? They're too large for this middle bond, and so it instantly breaks in half. When you break this 6-carbon molecule in half, you're going to eventually have twice as many halves as you had fulls. And so if we have 3 full 6-carbon molecules, now we're going to have 6 3-carbon molecules. These are called 3-PGA. Then we're going to bring in 6 ATPs and 6 NADPHs. Both of these came in from the light-dependent reactions. Okay, that was made in the thylakoid membrane. They're going to come in from that thylakoid membrane. ATP is going to bring in energy, and NADPH is going to bring in the 2 electrons and a hydrogen ion. Okay, those are going to be brought in from this taxi. Once the NADPH drops these things off, it will be oxidized to NADP+. That NADP+ can travel back to the light reactions. Once ATP drops off the energy, ADP will transfer or travel back to the light reactions so that it can be converted back to ATP. And this one can be converted back to NADPH. Once you add the electrons and hydrogen ion and energy to these 3 PGA molecules, you will convert the 2 or 6 total 3 PGA molecules into 6 G3P molecules. In the 3rd step, we are going to remove a G3P. So 1 G3P comes out. Those will be the G3Ps that we use to make glucose. If you remove 1 from our 6 total G3Ps, you now have 5 G3Ps. We now, in the regeneration of RUBP step, which is step 4, require an input of 3 ATPs, which are going to bring in energy so that we can shuffle the 5 3-carbon molecules and convert them into 3 5-carbon molecules so that we start and stop this cycle with Rubisco or 3 RUBPs. Okay, this is the Calvin cycle, also called the light-independent reactions, also called the dark reactions. Okay, if you have questions, which you may have, we expect you to bring those questions in class. We're going to obviously review this in class But for now, I think that's where we'll end it. Isn't the Calvin cycle sweet? See ya.